Welcome to the presentation of the poster PICKLUSTER, a protein interface clustering and analysis plugin for UCSF ChimeraX. I would like to start with the question why protein interfaces are so important. Protein complexes are key components in the majority of biological processes, such as the activation of receptor molecules, DNA replication, signal transduction, protein export and transport, recombination, and repair. In order to understand the underlying mechanisms of protein complex formation and macromolecular recognition, the identification and characterization of protein-protein interfaces is required. Moreover, protein interfaces can be targeted by drugs for protein complex inhibition. Now I would like to talk about the question why we've decided to implement something like PICKLUSTER and how it actually works. In order to investigate the interface, we need to know what part of the complex forms the interface and which residues are part of it. Protein interfaces can be really large, consisting of geometrically separated domains, posing a challenge for targeting the interface. So what PICKLUSTER does, it calculates the residues in the interface and divides the interface into smaller sub-interfaces, which could facilitate the targeting and helps defining the interface. PICKLUSTER starts with calculating the distance between heavy atoms, which means non-hydrogen atoms, of user-specified chains using a KD tree data structure. Heavy atoms within a distance of 5 angstrom are considered interaction partners. The center of mass of each residue exhibiting an interaction partner in the protein interface is computed in a third step. The clustering of the interface is performed by calculating the distance of these centers of masses in the interface. If the distance is below the distance cut off, which is by default 5 angstrom, the residues are considered to be in the same cluster. In a last step, a list of the residues in the interface is reported in the UCSF ChimeraX log and the clusters are colored. PICKLUSTER offers various analysis options for the calculated clusters. So PICKLUSTER automatically colors the clusters and reports a list of the residues in the clusters in the ChimeraX log. But moreover, the user can choose from different analysis options. The first one is to color the clusters in the sequence. For this, PICKLUSTER generates a sequence coloring format file that can be imported into the UCSF ChimeraX sequence viewer and can be also exported for further comparison. A second option is that PICKLUSTER can show the atomic interactions in the protein interface in the UCSF ChimeraX main window. This option offers a more detailed view into the interface. Moreover, PICKLUSTER also offers the option to display a list of interacting residues per cluster in the UCSF ChimeraX log instead of just showing the residues in the interface. PICKLUSTER is implemented to not only work on protein complex structures that are obtained by using experimental methods like X-ray crystallography or cryo-EM, it is also implemented to work on models generated by AlphaFold. For these models, PICKLUSTER offers different analysis options than for the structures. I would like to show the mentioned options and PICKLUSTER outputs for the example of the dimeric complex of UL15 and UL33 from Epstein-Barr virus. When applied, PICKLUSTER calculates 3 clusters in the protein interface that are here colored in purple, green, and pink. The clusters are calculated using a distance cutoff of 5 angstrom for the clustering. This is a list of all options provided by PICKLUSTER for the visualization and analysis of the clustered protein interface in models. This includes the coloration of the calculated clusters according to their PLDDT, which is a confidence measure of AlphaFold. Red indicates regions of poor confidence and blue depicts high confidence regions. What is already visible in here is that cluster number 2 shows a very high mean PLDDT and the error that is represented by the PAE is quite low, which indicates high confidence in the predicted interface cluster. Last but not least, I would like to sum up and conclude. So we developed PICKLUSTER, a plugin for the protein visualization tool UCSF ChimeraX that uses spatial properties for clustering protein-protein interfaces and provides a range of scoring metrics for the analysis of the results. It can be potentially applied for the detection of targets for ligand docking or the analysis of hotspot residues in the protein-protein interface. Moreover, it could be used for the functional characterization of mutations in protein-protein interfaces. Now I would like to thank you for listening, and if you have any questions, please don't hesitate to contact me via the chat option.